So yeah, I'm just going to get right into our data today. What we're trying to do, in short, is model the Berlin patient. And I think most of the people in the audience are pretty familiar with this clinical course. I'm just going to touch on a few of the aspects that we're interested in modeling. The first, of course, being the fact that Timothy Brown received a bone marrow donation from a donor that was homozygous for CCR5 delta 32, which is a well-characterized null mutation in the CCR5 co-receptor for HIV. In addition to receiving these cells, he also underwent a chemo and radiotherapy conditioning regimen. In the bone marrow transplant field, that's what's needed so that the donor cells have room to engraft. In the case of an HIV-infected inpatient, you can imagine that the total body irradiation and chemotherapy that he received would also kill a portion, but not all, of the latent viral reservoir. So we want to model these two facets in a stem cell transplant model that we've developed in the Pigtailed macaque. So I work in Hans-Peter Kiem's lab, and we have over a decade of experience using pigtails to model stem cell transplants, hematopoietic stem cell transplants, for a number of diseases, not just HIV. But in the case of our HIV studies, the goals very broadly here were to establish a model of infection and transplant using our expertise in the transplant end, and then the wealth of knowledge that's out there for NHP models of NHP and AIDS. Bringing those two together, what I'm going to show you today and really focus on is our efforts to edit the CCR5 gene in hematopoietic stem cells and what happens then when we transplant those autologous CCR5 deleted stem cells into animals, both SHIV-infected animals, and I'll get into what that means, and SHIV-naive animals. So just first to go over what our strategy is, this is a schematic of how our transplants work. We have, as I mentioned, juvenile pigtail macaques. We treat them with GCSF and stem cell factor, and what that allows us to do is harvest a large number of CD34 positive hematopoietic stem cells from bone marrow aspirates. We isolate the stem cells out of that bone marrow product, and we can culture them for a number of days ex vivo in the presence of the growth factors that you see here. While the stem cells are being cultured ex vivo and engineered, as I'll talk about in a minute, the animal's receiving a myeloablative conditioning regimen. And in our case, that's total body irradiation of 1020 centigrade. So myeloablative conditioning in this case means that if we didn't put the cells back into the animal wouldn't be able to recover without the reinfusion of these cells. And that's an important point I'll touch on again later in the talk. Or I missed the most important part. So what we're trying to do with these cells ex vivo is engineer them, as I mentioned, with deleted CCR5 alleles. And our strategy is to use zinc finger nucleases. Zinc finger nucleases are one of a number of nuclease platforms that you've probably heard a lot about, along with CRISPR-Cas9, as you'll hear about after me, as well as TALENs and others that are capable of targeting to a genetic locus of interest, inducing a double-strand break, and utilizing cellular machinery to induce a mutation, hopefully a null mutation at that site. We're using mRNA to deliver our zinc finger nucleases into cells by electroporation. After we culture the cells, deliver the nucleases, and let them recover overnight, we then put them back into the conditioned animal. So there's a lot of different metrics that we can use to ask the question of how well this is working. For the purposes of time today, I'm just going to focus on the last three and bullet point the first two. The first two are that, number one, we see normal hematopoietic recovery following these transplants. Again, we're giving them total body irradiation, which is going to ablate to a great extent their T cell counts, their total lymphocyte counts, and myeloid lineages as well. But the ability of those cells to recover after we put the stem cells back in doesn't seem to be impaired in these transplants. And we have a lot of historical data from other gene therapy experimentation to make that comparison pretty strongly. Well, another thing that we see is that we have multi-lineage engraftment of these gene-edited cells. What that means is that we're putting in true hematopoietic stem cells that are capable of making not just T cells, the protected T cells would be what we're most interested in, but also myeloid lineage cells that may or may not be susceptible to infection, and basically every cell type that we can look at, we see deletion of CCR5. But what I really want to focus on for today is what the cells look like ex vivo, basically what is the bulk amount of CCR5 disruption we see, what is the amount of CCR5 disruption on a per cell basis, and then what do the cells look like when we put them back into the animal. So this is the data from a cohort of 4 animals that we've transplanted. These were the first 4 that we got really representative results on, and I'll talk about more of the animals later in the talk. As you can see in the top, what I'm showing you here is these are CD34 cells that were left in culture while the bulk of the cells went back into the animal. And we make total genomic DNA from these cultured CD34 cells, and then use a deep sequencing strategy to quantify the number of disrupted CCR5 alleles. You can see it ranges anywhere from 30% in the lowest animal here to upwards of 60% in the best representation. But a question that you might come up with early on is, for instance, in this animal we see 50% disruption. Does that mean that half of the cells have both alleles disrupted, or does that mean that all the cells have one allele disrupted? In stem cells, we can address that by doing colony forming assays. So plate a very limiting number of these cells in a semi-solid media. Colonies grow out from those, and they should all arise from single cells, such that when we sequence each colony, they're either going to be 0% disrupted, 50% disrupted, or 100% disrupted. And it's these biallelic or indel events that we're most interested in quantifying, since those are the cells that hopefully will be null for CCR5 depending on the mutation we've induced. You can see that we have anywhere from 0.5% up to about 10% of those cells that are biallelically disrupted. One thing that I want to note is these are low, but in very early data that we have where we've challenged these animals with SHIV, this actually is the most promising animal that we're seeing so far. So I don't think the bulk amount of CCR5 or the bulk amount of biallelic disrupted cells is necessarily predictive of which animal is going to have the best response to SHIV. And I'll touch on that a little bit at the end. So again, the post-SHIV data is a little too early to show at this point, and what I really want to focus on is just the ability of these cells to engraft. So what we can see is these are the same 4 animals I just showed you, and this is what it looks like when we put the cells into the animal. That very early after the infusion, we see that we have peak disruption of up to 40%, depending on the animal, and then that balances out more at about 2 to 5%, depending on the animal. A couple things I want to highlight from this data are, number one, this is not surprising based on our gene therapy experience in the macaque. I think a lot of the cells that we're culturing ex vivo for various reasons lose long-term repopulation potential, so that when we put them back into the animal, they're not going to be able to last out this long. The other thing is that we're not clear at this point, just based on the markers that are available in the pigtail macaque, whether we're effectively delivering these nucleases to true hematopoietic stem cells. So those are 2 objectives we're very interested in searching out with the reagents that are available. So everything that I showed you so far is in animals that are uninfected. As I've mentioned, we've gotten through all the transplants of these animals, and we're very early in the SHIV response. But what I can show you in terms of infection data is using, as I've referred to a couple times, our simian human immunodeficiency virus, or SHIV, infecting the animals first with that, then suppressing the animals on antiretroviral therapy, and then performing the transplant. So this is more of a preclinical model where we can set up a representation of a patient that's been infected and is well suppressed, and then assess the feasibility and the efficacy of transplants in that setting. So just briefly to talk about the virus we're using, this is an envelope SHIV. So the greatest portion of the virus in white here is based on SIV MAC239. The envelope is HIV clade C from a primary isolate. And then the antiretroviral therapy regimen that we're using consists of 2 RT inhibitors and 1 integrase inhibitor. And we've shown in data that I don't have time to go over today that this virus works quite well. It infects the animals. We see virus in tissues as well as in blood. The suppression works quite well at the same time, and we really have a nice system set up to screen not only this CCR5 deletion strategy, but any protective gene editing or gene therapy strategy. So what I want to show you today for the post-SHIV data is just looking in vitro. All of the animals that we've infected, are all but one actually, that we've transplanted that were infected and ART suppressed, still remain on ART at this time, because we want to give the animals plenty of time for their immune systems to recover from the transplant before we take away the cover of ART. So what we can see in vitro, at least, is that this is the uninfected cohort that I showed you before, just averaged out. around 35 to 40% disruption and about 5% biallelic disruption. And then in our infected and ART-suppressed cohort, you can see a slight uptick both in the amount of bulk disruption at CCR5 and the number of biallelic disrupted stem cells. We don't think that this slight uptick has anything to do with the infection status of the animal. It just, I think, better reflects the fact that as we've done more and more of these transplants, we've gotten better at them, and there's a few tricks that we've learned that have animal by animal increased the efficiency with which we can disrupt CCR5. So just to conclude, what I've shown you in the data so far is that we can disrupt CCR5 and macaque HSCs with an efficiency approaching 70% ex vivo. In our most recent cohort of animals that we just transplanted a few weeks ago, we've just gotten data back that we're upwards of 80% at this point. So we're definitely improving with every animal that we do. Up to 10% biallelic editing is what we have right now, and multi-lineage and long-term engraftment of those cells is very clear from all the animals that we've done so far. So, of course, what we're most interested in at this point is characterizing the response of these animals to Shiv challenge, both the animals that were transplanted first and then infected, and the animals that were infected and suppressed and then transplanted, where we would be taking the antiretroviral therapy away. And as I've kind of hinted at a few times, we're observing a threshold effect. So the 4 animals that I've shown you where they were transplanted and then infected, we have data, very early data, suggesting that we are seeing a virus-dependent selection in some of the relevant subsets, but it's only in a portion of the animals, suggesting that where the levels to which we're deleting CCR5 now are sort of right at the borderline. And so I think that there are various ways that we could bump up the amount of disruption that we see, and that would be very advantageous. So just to end up, I wanted to go over a future direction that we're interested in pursuing, which is trying to actually knock in a selectable marker at the disrupted CCR5 locus. So as I mentioned, we're inducing these double strand breaks, and right now we're just counting on a mutation that is induced by non-homologous end joining to create a null mutation in that gene. But if we can select in a marker, in this case I'm using GFP just as an experimental surrogate. If we can select in a marker at that locus, then we would be able to potentially select out 4 cells that are disrupted. The way that we're knocking in our various markers is using a promoter with CCR5 or a promoter-driven cassette with CCR5 homology arms that's packaged into an adeno-associated virus. We can then treat the hematopoietic stem cells or transduce them with this virus prior to the zinc finger nuclease delivery. And then basically the template for this homologous recombination reaction is already in the cells when the double strand break is induced. And again, I'm just going to show you data for GFP. That's not going to be therapeutic in any way, but it allows us to have a nice, easy metric of how much we're able to do this. And what we can see is when we have our zinc finger alone, of course, we don't have any GFP positive cells, but when we do the same CCR5 engineering strategy in the presence of this AAV GFP donor cassette, we have 10% of the stem cells are now GFP positive. This is stable as far as we can tell ex vivo up to 9 days. We have some more recent data where up to a month we can see that that's a pretty stable integration event. And what we're interested in doing, again, is not just using GFP, but knocking in a selectable marker. Here I'm showing you a marker that we have a lot of experience with in Hans Peter's lab called NGMT P140K. This is, to make a long story short, a chemo selection cassette that allows us to select with O6-benzylguanine and BCNU and temozolomide. And when we do that, and these are from historical animals where they're actually stem cells are transduced with this selectable marker fused to GFP, you can see that whenever we give chemo selection, we get a very significant increase in the amount of gene-marked cells in the granulocyte lineage as well as the lymphocyte lineage. So based on the fact that we're at sort of this threshold right now, without this selection, we feel good that if we can knock in a selectable marker and actually bump this up even to a slight degree, we'll really jump over that threshold and get into some really productive responses with Shift challenge. So as I mentioned, just to wrap up, we're always interested in maximizing CCR5 disruption and evaluating the changes in Shiv in the animals following Shiv challenge. And on the other end, this selection strategy is something that we're pushing very hard on at the moment. So with that, I'd like to thank Hans Peter and the rest of the lab. As you can see here, it's a huge group effort to do these studies. I'd like to especially thank our animal technicians, Veronica, Erica, and Kelvin, who are instrumental for these studies, as well as Sangamo Biosciences, who developed the zinc finger nucleases that we're using, and within our Martin Delaney collaboratory, within which this work was done, the expertise from Shulock Hu for our NHP studies, as well as Keith Jerome. So thank you very much. I'll take any questions.
Just while we're waiting for people to come to the microphone. So you mentioned that the transduced cells, we saw that they kind of died away quite quickly after infusion, and you mentioned that might be because of handling, etc. If you have the GFP and you handle them more and select the GFP positive ones, will you just end up without repopulation, or without the other competitors, you think they'll get up to normal levels?
Yeah, the optimization of the knock-in is definitely ongoing. There's going to be a lot of different things to tweak in that aspect. I think as far as the loss of the cells, we just need to be able to better characterize true hematopoietic stem cells. And we're having to sort of develop the reagents from scratch to do that, because there's certain markers that are well known and characterized in humans that we haven't been able to use in macaques.
Okay, thank you. Well,
good work. Have you looked for off-target effects of the zinc finger nucleases? Are you planning to do it?
Yes. Yeah, so we've I didn't have time to show that data here, but we used a CellX-based approach, or Sangamo used a CellX-based approach to rank order 25 off-targets, which were all analyzed by deep sequencing. We actually need to sequence deeper, because to the depth that we analyzed all those sites, we didn't see any. So as far as we can tell right now, this is a very specific and on-target nuclease that we're using.
Okay, thank you very much.
Thank you.